In today's session, we learn about types of RNA, transfer RNA, and the ribosomal RNA. In the previous session, we learned about messenger RNA, discovery and its structure. Discovery of transfer RNA. Transfer RNA was first found to be discovered by Paul Zemanick. It was known as adapter molecule, which plays an intermediate role between transferring the message from the messenger RNA to the formation of proteins by linking on ribosomal RNA. Transfer RNA was first discovered using cell-free medium wherein all the nutrients required are provided with the addition of mature RNA. As you can see in the diagram, here the cell was subjected to lysis by detergent. Later, the cellular contents are transferred into a medium with the mature RNA, mature mRNA. The mature mRNA is formed by the process called splicing after transcription, which will be dealt in detail in the later sessions. These were added into a cell-free lysate medium, wherein the formation of peptide bond mediated by transfer RNA was observed. This was concluded by radio-labeling carbon, which was later found in the resulting peptide bond, and this protein was transferred to the microsome, an organelle present in the cell, and the radioactivity was detected in that particular organelle containing peptide chains of C14 content. That's how transfer RNA was discovered, which proved involvement of a particle or a component without the presence of cell, which mediates the information transfer between messenger RNA and aids the formation of proteins. Structure of transfer RNA. Here, as you can see in the diagram, a popular structure known as cloverleaf structure is generally studied, whereas transfer RNA also takes other structures known as L-shaped structure. In the process of mediating this translation, structure of cloverleaf model, it contains a D loop, anticodon loop, pseudo U loop, and a acceptor arm. Here, the anticodon loop is the one which contains the codon complementary to that of the codon present in messenger RNA. This codon is the one which has a specific amino acid coding for it. Each tRNA is specific. A single tRNA contains a particular anticodon which codes for a particular amino acid. The transfer RNA aids for the formation of peptide bond by attaching to particular sites on ribosome, which will be dealt in detail in ribosome in translation process. A glimpse of it include the ribosome has particular sites. Ribosomal RNA has a particular site in the smaller subunit. This is where the tRNA binds, and it facilitates the formation of peptide bond between two amino acids by bonding with consequent to tRNA molecule, which is facilitated by specific enzymes, thus aiding the formation of peptide bond. And after the formation, these tRNA leaves the ribosomal site, and other tRNAs come and occupy this site and propagates the formation of peptide. Here, as you can see, there is alternation between cloverleaf model and L-shaped structure during the process of translation, which takes upon the favorable conformation of the process to occur. The next one is the ribosomal RNA. As I've mentioned earlier, how the tRNA have specific site for them to be bound to the ribosomal RNA during the process of translation. Here you can see the mRNA has a codon, for example AUG, which codes for methionine. The tRNA will have anticodons for that, that is UAC, which links to the codon present in the mRNA exactly, and another tRNA occupies the subsequent site and aids for the formation of peptide bond, that is the covalent bond, and facilitates peptide bond, that is the bond between the N terminal and the C terminal of the consecutive amino acids, and facilitates the formation of particular amino acid. Example, methionine here. Methionine, if it is preceded by another amino acid, so say isoleucine, those two amino acids are formed. Later, the peptide chain formed is released through another terminal, aiding for the formation of peptide chain. As you can see, the ribosome consists of a larger and the smaller subunit. The larger and the smaller subunit exists separately in the cell, and it only assembles during the process of translation by signaling and chemical stimulations. This ribosome is generally found, ribosomal RNA is generally found with the ribosome, which is in turn present on the endoplasmic reticulum. Discovery of ribosomal RNA. This was first discovered by ultracentrifugation technique, as you can see in the diagram. When the ultracentrifugation was performed, there was a different sedimentation carried out. That is, the subunits of ribosome, as you saw earlier, larger and the smaller subunits, sedimentated separately. This gave an idea that there is an organelle called ribosome which contains the ribosomal RNA, and it sediments separately and it assembles only when there is synthesis of protein, that is during the process of translation. This can be performed even today. The sedimentation coefficient is known as Svedberg coefficient. That's why the S is given for every subunit name. That is 50S subunit, 30S subunit. In case of eukaryotes, it is known as 60S subunit and 40S subunit. Here, types of ribosomal RNA. Here, as you can see in the diagram, in eukaryotes, ribosomes are divided into larger and smaller subunit like the bacterial rRNA, but in bacterial ribosome, it is divided into 50S and 30S subunits, totally known as 70S, although many of them have doubts regarding why it is called 70 and not 80. That till now people are trying to find answers for that. But as of now, 50 plus 70 in biology becomes 50 plus 30 in biology becomes 70, as in case of ribosome. In case of eukaryotes, 60S is the largest subunit and 40S is the smaller subunit. This larger subunit is made up of multiple components, that is 5.8S rRNA, 5.5S rRNA and 28S rRNA, composition of the larger subunit. The smaller subunit is made up of 16S rRNA, and the remaining 33 proteins. The same goes with the prokaryotic ribosome, which consists of the 50S and the 30S subunits. The 50S here is made up of a 5S rRNA and 23S rRNA, and the remaining constitute for the proteins. And the smaller RNA is consisting of 16S rRNA and 21 proteins. In case of eukaryotes, 18S rRNA and around 33 protein components. This is the composition of prokaryotic and eukaryotic ribosomes, which is very important to be known. Thank you. If you find this video informative, please do like, share and subscribe to our channel. If you feel you need to know more about it in detail, please check for the link below in the description for detailed notes.